The overall goal of this procedure is to analyze whole zebrafish embryos between the 4-cell stage and 32 hours post-fertilization for the presence and location of apoptotic cells. This is accomplished by first dechlorinating embryos at the desired time point. Next, the embryos are fixed and permeabilized. Then, the embryos are incubated with rabbit anti-activated Caspase 3 antibody, followed by a fluorescently tagged anti-rabbit secondary antibody to fluorescently label apoptotic cells. Finally, the embryos are analyzed by fluorescence microscopy. Ultimately, results can be obtained that show differences in number and location of apoptotic cells between control and treated or mutant embryos through immunofluorescence.
The main advantage of this technique over existing methods, like analysis of apoptotic cells by acridine orange staining or the tunnel assay, is that the Caspase 3 assay is a highly reproducible and relatively inexpensive method for experiments in which photographic documentation and/or quantitation of apoptotic cells is required across multiple samples. Demonstrating the procedure will be Shelly Sorrells, a graduate student from my laboratory, and Cristhian Toruno, a technician from my laboratory.
Under a chemical fume hood, prepare 50 milliliters of 4% paraformaldehyde, or PFA, by adding PFA powder to 1x PBS. Shield from light and stir at 60 degrees Celsius for 1 hour to dissolve. Store at 4 degrees Celsius for 1 month, or at minus 20 degrees Celsius for 2 years. Prepare 100 milliliters of 4% methylcellulose by heating 30 milliliters of water to 80 degrees Celsius. Then add 4 grams of methylcellulose and stir to dissolve at room temperature.